Okay, so a few housekeeping things. First thing that some people have gotten in contact with me about, people are starting to get nervous because our exam, as I just mentioned before I hit the record button, our exam is a week from tomorrow. Now, this exam covers chapters 1, 2, 3, 4, 5, and chapter 8. It's not chapters 1 through 8. We are not doing 6 or 7 at this time. Chapters 2, chapter 5, and chapter 8. So 3 out of those chapters are completely review from Bio 1. And if you've already taken chemistry, chapter 2 is a review from chemistry. Chapter 5 is all about eukaryotic cells, which you should have covered almost in its entirety in Bio 1. The part that I particularly don't get to with my classes, as well as I know some other instructors don't get to, the fungus and the protists, we're going to go through that part of the chapter 5, but the beginning part of chapter 5, you just need to review that on your own. And it's same old stuff, all the different parts of the cell and their functions of the eukaryotic cells. So this should not all be new material for you. And if it is, if you didn't have Bio 1 or you haven't had it for a while, yes, it's going to take you longer to prepare for this test than someone who just came out of Bio 1. Absolutely. But it is different for everyone. Now, those of you that have been in to Canvas, you'll notice that I did post a second set of Connect homework for chapters 1, chapter 2, and chapter 3 I put in 2 parts. The first part is just about the 5 eyes, and then the second part is about the microscopy and staining, all of which we've already taken time to do. I've added a microscope lab. It's completely virtual. It is completely virtual, which means you need to go through Connect, get to this exercise, and there'll be a woman head there who talks to you and goes through a basic review of how to use a compound bright field microscope. And then it's kind of cool. I sat and did a lot of it yesterday. It goes through how to do immersion oil procedure, and it has you you have to click on the dropper bottle, put a drop of oil on, change your objective by clicking on it, lowering the microscope so that you can focus. And then I think you have to answer a few questions. I believe there are places you can save it along the way. It's just watch, and when she says you can save and come back later, I would always do that if I were you. And then I believe at the end there are some questions. I didn't make it all the way through the end myself. It takes a while. I would set aside a good an hour to go through that. That will help us for when we get our kits, because there is a microscope in your kit. And then when you actually go to use it, you will have this already available that you can review and refresh. So I added those 3 things. I'll be adding homework for chapter 4 from Connect. From chapter 5, that one will be short because I'm only going to add, actually I'll leave it long, but I'm only going to, that's again, it's for review. It should go very quickly. And then for chapter 8, I probably won't put that homework in until Wednesday, until after we've covered chapter 8, which is metabolism. We will not be doing photosynthesis if you're reading ahead, but if you aren't reading ahead, then you can already know that we're not going to be using that last section of photosynthesis in chapter 8. But the rest of it about the Krebs cycle and enzymes and all of that should be familiar to you, electron transport chain. And that will be covered on the first exam, which is a week from tomorrow. So you still have 8 days. If you haven't done anything, you still have 8 days, but it will take you a significant portion of each of those days to get all this stuff done. So I wanted to tell you that. And then I wanted to discuss the last bit of chapter 4. And then after I'm finished with doing 4 and the parts of 5 that we need to cover, I'll talk to you a little bit. I know some of you are having difficulty with the lab kits, with the purchasing of the lab kits, and there is money available from the college, but you have to fill out the forms, and I can try to help you find them, but I don't have those forms myself. You'll have to contact someone in student affairs, or your advisor would be able to help you with that. So let's take a look at the rest of chapter 4 here. Can somebody clue me in where we left off? I seem to remember we left off at the, I think the last thing we talked about was the different types of cell walls. Is that true? Yes, cell walls, end of cell walls, I think so. All right, so, okay, thanks, Bridget, I appreciate it. So I'm gonna put this here or over even further. And I'm gonna get rid of the picture of polysaccharides from my BIO1 class. And I'm gonna put superbacteria up here real quickly, kind of over here on the side. Bring you guys in a little bit tighter. So hopefully it'll focus a little better on the rest of the board. Okay. Atypical cell wall, okay. All right, so if we have our friend superbacteria here, which we talked about last time with his flagella and all the fimbriae sticking out of it, right? And the black line that I've drawn is going to be the cell membrane. Outside of that cell membrane, if you're looking at that picture on page 83, right? Outside of that, we would have this, right? So here's cell wall right up against that. And remember the cell wall, the main two types of cell wall that we have are the gram-positive cell wall and the gram-negative cell wall. Right? And we talked about the differences between gram-positive and gram-negative. The gram-positive, just a quick review, the gram-positive cell wall is going to be made of peptidoglycan, predominantly with some teichoic acids and lipoteichoic acids. The gram-negative cell wall is going to contain an outer membrane and then a thin layer, 1 to 2 layers of peptidoglycan, and that's going to comprise the cell wall. If you are a mycobacterium, you're going to have mycolic acid and peptidoglycan in that cell wall. If you're a mycoplasma, you don't have a cell wall that has peptidoglycan in it. Instead, you just have that mycolic acid and I'm trying to think what else. Mostly mycolic acid, that waxy component. Sorry, it's the best I can do. I've got the only thing I have is the audio from my computer. So if it's not working very well, I don't know what to tell you. I'll try to not be below, I don't even know where the microphone is. All right, so outside of the cell wall, then remember, sometimes bacteria have that other layer. Looks like it doesn't work. Or green. Okay. So outside of that, we would have glycocalyx or S layer, right? Slime layer or a capsule or the glycocalyx. And that's just to refresh your memories from last time, because today we're going to start talking about the cell membrane or the plasma membrane and go inward into the cell from there. So this all right, so cell membrane, all membranes in all cells, you need to remember this from BIO1, they are all a phospholipid bilayer. Which looks something like this, right? With the phospholipids, with the phosphate heads that are negatively charged, and the lipid tails that have that little kick on them because they have a double bond there in their lipid chain, their hydrocarbon chain. These we call hydrophilic heads because they like to be near water, and the hydrophobic tails of the molecule on the inside of the membrane there. So phospholipid bilayer, 2 layers of phospholipids with water inside and outside. And then of course, in a bacterial cell, outside of that, here, and I'll just kind of draw it like this, outside of that is going to be where that cell wall is going to be composed of. Right? So I would remind you, let's structure of the cell membrane, and more importantly, the function of the cell membrane is to be a selectively permeable barrier. And again, if you just had Bio 1, this is chapter 7. If you haven't had it in a while and you still have your book, I'd go back and review it. Selectively permeable means you can, some things are going to be able to travel in and other things are not, and some things are going to be able to travel out, other things are not. Remember, there's both passive and active transport through this membrane, which we will talk more about in chapter 7, which is not included on this exam. Now, there's an additional important function of the cell membrane in bacteria that's not the same as in your cells or in eukaryotic cells. And that function, that second function, is that it's also the location, or we say the site, of metabolism. So, and particularly what I'm referring to is for metabolism is if this bacterial cell happens to be a photosynthetic bacteria, it's going to be instead of the thylakoid membrane. If it's a bacterial cell that just uses sugar, uses glucose, sort of like your cells, what we mean by location of metabolism is that I mean the electron transport chain is that would normally be, we would think of in eukaryotic cells inside the inner membrane of the mitochondria. There are no mitochondria in this cell. Remember, no membrane-bound organelles. So the only membrane they have to use for transporting hydrogens across the membrane so that ATP synthase can pump them through and make ATP is the cell membrane. So this is an important new function of the membrane that wasn't a function we probably learned about before. Now we're finally through all the coverings of the cell, and we can talk about what's inside. Okay, so contained in the cytoplasm, right? So that doesn't go with that, but the next thing, they were numbered on your handout, but I don't know what numbers they were, and it's unimportant, but I probably shouldn't put that right next to that. Let's put it over. Cytoplasm. So the cytoplasm is similar to the cytoplasm in your cells. It's mostly water and all the other stuff that's inside the cell, right? So water is this, what we would call it, is the solute in this case, right? Solvent, sorry, is the solvent of the cytoplasm. So full of water and then full of some other stuff. This is where the DNA is located, where the chromosome is located. I'm going to make a list so I don't forget anything. This is where plasmids are located if the bacteria has a plasmid. This is where the ribosomes are. And another feature that's present usually only in bacteria, something called an inclusion body. Inclusions, you'll see them called, or inclusion bodies. I think that's all we need to cover that's in the cytoplasm. So let's talk about the chromosome first. I think we've already mentioned this when we were talking about that bacteria have a chromosome that is a single chromosome. I'm going to draw it really large because that's how it kind of looks in your book. Right? We call this the nuclear area. It's a single, if I stretched this out, it would just be a single loop like this, right? And I would draw it kind of like that. And I usually put lines through it to indicate that it is still double-stranded, double helix, same basis as your chromosomes. And then if I were to draw this as that single chromosome, right, it would be in a much bigger cell, like that. In reality, is more real how your book has drawn it, in that that chromosome takes up most of the space inside the cytoplasm. It's quite large, and only if you took it out and stretched it out could you see that it was a complete circle. So it's a single chromosome, one chromosome. It is circular, and it contains, E. coli contains about 4,000 genes. Relative to you, which you contain like 30,000 genes. It's double helix, double-stranded, A's with T's, C's with G's, all the rest of it's the same. So that's one thing that's inside. And of course, the function of the chromosome is that it holds all of the genetic material for the organism. It has the code to build all the proteins, and then the proteins, of course, do all the work of the cell. Now, sometimes, so a plasmid is a sometimes, some bacteria have plasmids, and plasmids are smaller extrachromosomal pieces of DNA that are also circular. I'm going to draw kind of badly here. This little brown guy is a plasmid. You guys can see that. I don't have space to write that out, so I'll say it much slower so you guys have a chance to write it down. A plasmid is extrachromosomal, all one word, meaning it's outside of the chromosome. The extrachromosomal piece of DNA that is also circular, double-stranded, double helix. Why do they have those? Well, the cool thing about plasmids is that if you have one, I like to call them bonus DNA. Bonus, put it in quotes. Bonus genes. So these are characteristics, this is DNA, the code for proteins, that if you don't have them, you can still live perfectly fine. But if you have them, it makes you much more likely to be able to cause an infection. So, for example, if you have a plasmid, what kinds of genes are on that? Well, one thing that might be on it is like antibiotic resistance. Another thing might be the ability to make fimbriae. Another thing might be the ability to make a capsule. Another thing might be able to make a toxin that can hurt the host and allow the bacteria to multiply more. So see what I mean by bonus? It's better for the bacteria, worse for the host, right? In this case, that would be you, if we're talking about a human bacteria, a bacteria that affects humans. Ribosomes are ribosomes. You've seen those before, right? Ribosomes in bacteria are a little bit smaller than in eukaryotes. We call them 70S ribosomes. And they have two components. They have a large subunit and a small subunit. Right? The one that's larger is the large one, the smaller is the small one. And those of you that remember from BIO1, that remember protein synthesis, that's the function of the ribosomes, to make the protein. Try to remember proteins do everything in cells, so we need to make a lot of proteins. So bacteria are going to have lots of ribosomes. They're all free, floating around in the cytoplasm. And remember, ribosomes do not have a phospholipid bilayer around them. They are not membrane-bound organelles. So I'm going to draw some ribosomes on my superbacteria here. And I'm drawing them probably larger than I should. Usually in pictures, they just show them as dots. Right? So the picture in your book is just going to show it as a dot. But I'm trying to make it look like something. Function, protein synthesis. Right? They're a different size than yours. That's a 7-0. And the reason for that, that's important that you note that, is because later when we talk about antibiotics and drugs that affect bacteria but don't affect the host, this is going to be one of the targets, because if you can stop the bacteria from making the protein, you can stop them from growing, you can stop them from causing an infection. And then inclusion bodies, those are also sometimes, so some bacteria have them. And we call them storage areas of a particular, either a nutrient, like a small organic molecule, or sometimes they're actually an element or a combination of elements of a compound. So I'm going to draw a big yellow inclusion body here and label it sulfur. Right. So some bacteria, for example, use sulfur for their metabolism, and so they collect up sulfur and it kind of forms this globule inside the cell. Sometimes it's a group of fats, if the bacteria can use that for metabolism. I think there's an awesome picture in your book of a bacteria that actually has inclusions of iron oxide, which is what makes up a magnet. And pictures to show. So the bacteria is like this, and then it has these, they call them magnetosomes. They're inclusion bodies of iron oxide, and so they're always lined up, and then this bacteria, those always point to the north, like a little mini compass. So again, inclusions are sometimes, they're much more common in environmental bacteria, not so much in pathogenic bacteria, like the ones that cause disease, but the ones you find out in the soil, because they need to accumulate sometimes some of their nutrients if they're not very plentiful in the environment. And they can be used for identification purposes. Okay, membranes, cytoplasm. I'm going to take a quick check to the book to see that I've covered everything. And then I'm going to finish up. I think there's just one or two more little things to talk about in this chapter. I think I'm actually just going to show you guys the spore pictures that are in your book. So let's come back over here. All right. So down here in the PowerPoints from chapter 4, very end of these PowerPoints, there is a picture of endospores. This is a picture of the yellow is the DNA inside of a bacteria, and you can see most of these are dividing, which is why there's 2 zones of yellow in here. But this would be an example of there's like a concentration of DNA and then a little bit more of it over here to show you how much of it takes up inside the cell. Show the ribosome. There's some great inclusion bodies that have been stained. All right, so it actually pushes like all the DNA and all the ribosomes almost out of the way. And then let's talk a little bit about spore formation. We have a picture of that. Apparently there's also cytoskeleton in bacteria now. Guess they don't give us that great picture. All right. It's hopeful. All right, I guess I'll have to draw it. Dang it. All right. Give me a second. I'm going to erase, and then you are supposed to read the whole chapter and then focus on the parts that I'm specifically talking about. But I would feel comfortable, especially in these chapters, asking you pretty much anything. Because those beginning chapters, remember, chapters 2, chapter 4, chapter 2, chapter 5, and chapter 8. Those are all things that you should have learned in BIO1. So those are fair game for me to ask you in this class because you're supposed to remember them and know them before. All right, so the last thing that we need to talk about from this chapter is a little bit about endospores, which are awfully cool. This is a sometimes. Not all bacteria have them. These are for survival. So this is not reproduction, all capitals, not reproductive. So the word you learned in BIO1, spore, for a fungal spore or for a spore made from a plant, not the same spore at all. Those were for reproduction. These are for surviving harsh conditions. So here's our bacteria. And again, this is a sometimes. Some bacteria can do this, not, and I'm just going to put some, I shouldn't even say sometimes. Some bacteria can do this, most bacteria cannot. They tend to be bacteria that do live in the environment. So here's our bacteria. La, 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 living its life. And here's its DNA. So now I'm going to draw it more, how I will more commonly draw the DNA inside a bacteria like this. Yeah, so there's its DNA and its ribosomes and all that stuff, right? This we call a vegetative cell, actively growing. Means it's feeding and dividing like regular. When the conditions become harsh, and what usually indicates harsh conditions or stress for the bacteria, if I had red, I would use it, right? But I don't apparently. So bacteria is getting stressed out. So for you, of course, stress is a lot different. You have a test, you're late for work, whatever it is. For a bacteria, the stress is usually the amount of water that's available, like a lower amount of water than normal, so the bacteria can't do its metabolism. Temperature, again, if I had red, I would use it. Too hot or too cold, those are the biggest stressors. And when the bacteria experience those stresses, if it is a bacterium that can produce a spore, something happens. What happens is this bacteria begins to form what we call an endospore. And first thing it does, it's going to copy its DNA. So this was the color I was using for the DNA, right? As if it was going to divide. So it makes a copy of its DNA. And then it starts to produce what we call a spore coat. It starts to wall off this piece of DNA, this newly synthesized piece of DNA with a few ribosomes and a few essentials. And it forms inside. So the blue is called the spore coat. It makes this super, super, super resistant structure that we call an endospore. Endospore because it's inside the cell, right? Inside the vegetative cell. As these stressful conditions continue, the vegetative cell dies. And all that is left is the spore. And then we don't call it an endospore anymore. We refer to it as a free spore. So I'm just going to draw it like this. And give it a little DNA in here. All right, so this is a free spore. And this free spore is super resistant to environmental conditions. It's super resistant to heat. Can't kill it with heat. Most chemicals like bleach won't kill it. Alcohol won't kill them or destroy them. So, but it's like a resting or dormant state, not actively doing metabolism. It's not actively getting any bigger. It's just in a resting state. So it can survive ice. It can survive boiling, as I mentioned. Most chemicals, and it just sits there dormant until when? Until conditions improve. And when conditions improve, the free spore, it usually has mostly to do with water. The free spore will, kind of like those little toys that you buy for your kids at the grocery store. You take them home, you put them in water, and they expand, right? It expands into a vegetative cell capable of doing regular metabolism. I should have made this purple, not blue. Right, so making it purple. When conditions improve, VE, it goes back to being a vegetative cell. One vegetative cell, one endospore, one vegetative cell. Not reproduction. I'm not making two cells. Same cell. One cell forms a spore, and then that spore becomes one cell. right? And again, this is sometimes this is only certain bacteria can do this. And as I mentioned, most of them, mostly environmental bacteria. So bacteria that live in the soil, particularly, are spore formers. Right? This is a characteristic that you can stain for. If you remember back to chapter 3, we had cells, we had an endospore stain, as well as it's a characteristic that can be used diagnostically. If the bacteria is a spore former, you can usually see the spores when they're in this state or when they're free, like that, under the microscope. So we'll be able to see them. I don't know if we're going to have a spore slide, but I'm sure in the virtual labs there's some spore slides available somewhere. And the last little bit of this chapter is a little bit about archaea, which are the bacteria, the other whole category of bacteria. I'm just going to take a whiteboard for this. So remember that they make up their whole own domain, right? So we have archaea, and these bacteria come, there's really more kingdoms than your book is telling us. I think there's 5 now, but your book goes through about 3 of them. They are the methanogens, the extreme thermophiles, and the extreme halophiles. So thermophiles is pretty self-explanatory. These are bacteria that are heat-loving, so they grow and thrive at temperatures above 80 degrees Celsius, which is really hot, 80 Celsius. And the extreme halophiles, these live in, a halophile is a salt lover, so salt-loving bacteria. So really high concentrations of salt, as high as 30% salt, or various salts. It could be sodium chloride, but it also could be other forms of salt. The domain archaea and the domain bacteria, which used to be called the domain Monera, or the kingdom Monera, right? These are all the bacteria that we are going to be learning about mostly. This contains about 5 different kingdoms. And I think your textbook has a picture of that. I think we saw that back in chapter 1 or so. But 1, 2, 3, 4, 5, 6, 7, 8. Your textbook is listing 8 kingdoms. So I'm going to put 5 kingdoms to 8, depending on what book you read, right? And then the third domain, of course, is eukarya. Oh, sorry. And this, of course, is everything else, right? These are all the eukaryotes. Now, what is the big distinguishing characteristic of all 3 of these domains? It's mostly based on ribosomal RNA, differences in ribosomal RNA. And there's a little chart in your textbook toward the end of this chapter that also talks about different types of ribosomes themselves, the ribosome size. So remember, we talked about bacteria having a ribosome of 70S. Archaea also has 70S ribosomes, and these guys, of course, are also domain bacteria. The domain archaea are also prokaryotes, right? Ribosomal size, and I guess I can't enter that same box. Oh, sorry. Third thing that I wanted to put on here was something about their DNA replication. is different between all three of them. And those are the main characteristics that separate them into the three big domains, right? And that's kind of the very end of this chapter, chapter 4. And the only other thing that I really wanted to show you was this picture from an older edition of the book that is actually in your in the PowerPoints that I gave you for this chapter. I'm going to have you take a look at this picture, because I think this picture does a little bit better job than what's currently in your textbook as far as these are the morphologies. These are the different arrangements of those morphologies, especially if we're looking at coccus and bacillus here. So see how you can see diplococci, right? And then these tetrads or these clusters or the chains, like we mentioned, cocci and chains, streptococcus, cocci in a regular cluster, staphylococcus. And then it shows a lot of variations of what a bacillus shape can be. And then it's a much better picture than I drew of the different forms of vibrios, spirilla, and spirochetes. So I did want you to pay attention just so you could see what different things look like, because it's different than, a little bit different, a little bit more detailed than how I showed it in my drawing. And that is the end of chapter 4, quite honestly. So I'm going to stop my recording.